Mm, so good. Carbohydrates, one of my favorite things in the world. So today we're going to talk about carbs. What are they? Where are they? Where can I get them? Chips, one of our favorite examples of carbs. A lot of people think it's breads, potatoes, grains. But today when we talk about carbs, I want to think about other examples in the body, and then also some that students forget exist, like carrots, potatoes. Our vegetables are considered carbohydrates as well. So today we're going to learn a little bit more about carbs and see if we can answer some of those questions. So first off, what are carbs made up of? Carbohydrates will be made up of carbon, hydrogen, and oxygen. So from before, we know that anything organic has to have that CH bond. So since carbohydrates have carbon and hydrogen present together, they're going to be considered an organic molecule. Specifically, these will be called macromolecules because they're made of many molecules and they're going to be larger. So within our carbohydrates, we can see that CH and O being present when we look at the structure of them. So this is called the nomenclature, basically the picture of the chemistry of the carbohydrate. So within this, you can kind of highlight over and see, I can see the C's, the H's, the O, the carbon, hydrogen, oxygen, and how these guys are formed. So usually our simple carbohydrates will either be a pentose or a hexose in shape. So pentose, think of a pentagon having 5 sides. So we can see 1, 2, 3, 4, 5. So fructose is an example of a carbohydrate that is a pentose structure. Hexose would be my favorite molecule, glucose, where we can see it has 6 sides, 1, 2, 3, 4, 5, 6. So those are some structures and some elements just to look out for in order to visually classify our carbohydrates. So our purpose of carbohydrates is living things will use them as a short-term energy source. So when you think short-term, I want you to think 24, 48 hours. It's not something that's going to last forever. So a lot of times you'll hear about athletes carb loading before an event. So they'll eat pasta the night before, and the reason for that is that that energy is going to be used in the next 24, 48 hours in order to give them the most energy possible to compete. Plants and animals also use carbohydrates for structural purposes. So with plant cells, we have the cell wall around the cell, which you can see I'm highlighting with my mouse there. And that's going to be made up of a polysaccharide called cellulose. So that's why when we eat our vegetables and our broccoli, that's going to be a polysaccharide because the cellulose is using structure in the plant cell wall for the broccoli. Other examples would be our insects. So the exoskeleton of insects, if you've ever seen a beetle or a cockroach, you can see that outer layer is going to be really tough. And that's that structure called chitin that helps protect the animal from being injured. So within our carbohydrates, we have 3 different levels that we focus on. We have monosaccharides, disaccharides, and polysaccharides. So mono, meaning one, saccharide meaning sugar, so literally translated as one sugar, is going to be our most simple form of carbohydrates. So glucose, we see that within our photosynthesis equation, simple sugar used for short-term energy. Fructose, you might have heard of before on commercials where they say, oh, this contains high fructose corn syrup. It's so bad for you. Fructose is basically just a sugar that they put to make it sweeter. So I always say everything in moderation, but we can see this fructose is a pentose shape. And then our last one right here, I didn't put the picture of the actual sugar, but we have deoxyribose, which you may have heard of in deoxyribonucleic acid, also known as your DNA. So later on, we'll look at how that sugar plays a role in the structure of your DNA, which will in turn aid in your genetics. Our second is called our disaccharides, di meaning 2, saccharide meaning sugar. So carbohydrates made of 2 sugars bonded together. So in this picture, I can see my glucose and my fructose, and they come and bond together through this glycosidic bond, and then they form a disaccharide. After our disaccharides, we have our most complex type of carbohydrate, which will be our polysaccharides, poly meaning 3 or more. So the main examples we focus on in biology would be starch, cellulose, glycogen, and chitin. So cellulose, that's going to be in the plant cell wall for structure. Starch and glycogen store our energy for a very short period of time. But here with our complex carbs, we can see many of those monomers or monosaccharides linked together. So this over here is actually what a picture of glycogen would look like. And each one of those little dots is a single monosaccharide bonded together. So we can see they're very, very large. And this picture on the left doesn't do it justice, but I just wanted you to see how it bonded together. So how do we go from our monosaccharide to our disaccharide or a polysaccharide? Great question to ask. As scientists, we're always asking questions. So we have this process called dehydration synthesis. So D means to remove, hydrate. We always think of water. And then synthesis, we know from Stringer, means to make or to create. So dehydration synthesis is literally the process of taking H2O, that water molecule, and removing it from two molecules to form something larger. So here we can see just two different examples of dehydration synthesis. On the bottom here, you can see with our carbohydrates, I have one monosaccharide and another. I remove an H and an OH. We can see now we have that disaccharide bonded together, and that water is going to be excreted as a waste. So that's a dehydration synthesis. But also we'll have our polysaccharides, and we're going to need to break those down and use them in our bodies as well later on. So I talked about our marathon runners earlier. They're going to pasta carb load that night before an event, and then their body is going to be doing hydrolysis in order to break that complex carb down into those simple molecules in order to use them on race day. So this is going to be the opposite of dehydration synthesis. Instead, it's hydrolysis. Hydro, water, lysis, to cut or break. So hydrolysis means we're going to add water to physically break a molecule. So down here, this image is showing actually a protein molecule. And we can see the water, H2O, is being added, and hydrogens are going here, and oxygen is there. So hydrolysis, exactly the opposite of dehydration synthesis. We're going to add that water to break that molecule in half. So here's the same example I showed earlier, except opposite. I have my disaccharide. We add water to end up with our two monosaccharides. So that's how we kind of use our carbs, by either building them or breaking them down. Just some hints for carbohydrates for identifying them. Usually carbs end in ose. There are some exceptions like glycogen, starch, and chitin, but the majority will end in ose: glucose, fructose, lactose, maltose, ribose. So that's just going to be a key thing to look for when trying to identify what carbs are. And then our last thing I want to talk about today is our indicator test for our carbohydrates. So for monosaccharides, we use this thing called a Benedict's test. So it's going to start off this light blue solution, and you'll add it to your samples. After that, you're going to heat it in a hot water bath for about 3 to 5 minutes, and you're going to look for this color change. So usually, at least what I test in my classroom, is you're going to see that change to orange. But sometimes, depending on what you're testing and what solutions you're using, you could get that green or red. Disaccharides actually have no tests whatsoever. So we'll skip over to our polysaccharides, our complex carbs. And for this, we use iodine solution, which is going to start off, I always say it looks like this rusty yellow color. And then you're going to add that to your solutions, and you're going to see an immediate reaction right away. And it's going to change to this black, dark color, and that will be a positive test for polysaccharides. So thanks for joining today to talk about carbohydrates with Mrs. Esser, and I hope I answered all your questions.